Good afternoon, everyone, and welcome to our Thursday afternoon webinar. I'm Melinda Bikini, the Chief Patient Officer with the Cholangiocarcinoma Foundation, and I'm also a 14-year survivor of stage 4 cholangiocarcinoma. We are so happy that you have joined us today, and we are glad to be joined by two special people, and I have their introductions coming up here. So we have Dr. Carol Addy, who's the Chief Medical Officer at Genfit, a France-based biopharmaceutical company with a mission to develop novel therapies and diagnostics for rare and life-threatening liver diseases with high unmet needs. We also have Dr. Mark Yarshawin, who's an associate professor of oncology at the Johns Hopkins Sidney Kimmel Comprehensive Cancer Center in Baltimore. He specializes in the care of patients with gastrointestinal cancers with a clinical focus on cancers of the liver, including hepatocellular carcinoma and biliary tract cancers. Welcome, both of you. And today, we're going to be talking about the phase 1b/2a study in patients with advanced KRAS-mutated cholangiocarcinoma. And I'm going to turn the presentation over to Dr. Addy, but I want to let the audience know that if you have questions along the way, please put them in the Q&A or the chat box, and we will get to them at the end of the presentation. Thank you guys for joining us. Go ahead, Dr. Addy.
Melinda, thanks very much for the warm welcome and for this opportunity. And let me just again introduce myself. I'm Carol Addy. I'm Chief Medical Officer at Genfit. Just to tell you a little bit about what we will be covering today, I'll start with just a high-level overview of Genfit, as I'm sure many of you have not heard of Genfit previously, after which I'll be handing over to Dr. Yarshawin, for whom I'm grateful for his joining me on this webinar today. And for those of you who may not be familiar with clinical trials, just to give you a high-level overview so that you have a fundamental understanding of the conduct of clinical trials before we go then into more detail about cholangiocarcinoma, the disease state, and then importantly, as is important for the context for the clinical trial that we are conducting, really understanding the science behind the investigational drug combination that is under investigation in this study. And then we'll be talking more, of course, about the details about the trial itself and wrapping up, as you said, Melinda, with questions at the end. So please make note of your questions so that we may ensure that your questions are addressed by the conclusion of the webinar. So just by way of high-level overview, as Melinda said, Genfit is a France-based biopharmaceutical company. Although 90% of our workforce is in France, I'm happy to say that about 10% of us are working here in the United States. That includes myself. I'm in the greater Boston area. Importantly, Genfit has now what is nearly a quarter of a century focusing on the development of novel therapies and diagnostics specific to liver diseases. And going back to the time when Genfit was founded, this resulted in the discovery of our first drug, elafibranor, fast forwarding to just last year, where we had very favorable data reading out in a pivotal trial for a rare liver disease called primary biliary cholangitis. Now, this is a truly exciting time for Genfit because the data for elafibranor are currently under review by regulatory authorities, including FDA, and we're anxiously awaiting what could be ultimately the approval of elafibranor in the coming weeks. So stay tuned there, clearly very exciting, but importantly, thinking about where we are now, really pivoting with a focus now on liver diseases that are rare and life-threatening. And this includes a pipeline of drugs that we are developing in a condition called acute on chronic liver failure, which is a final common pathway for many chronic liver conditions that result in cirrhosis, end-stage liver disease, and need for liver transplant. And this is complemented by what we'll be talking about today, another rare liver disease for which there are significant unmet needs. And we'll be talking about our development candidate, GNS 561, and the clinical trial, the phase 1b/2a trial that is now underway. And so with that, I will be passing to Dr. Yarshowan. So grateful for your taking the opportunity to join us today for this webinar, Dr. Yarshowan. And I'd be happy to advance the slides for you when you're ready.
Perfect. Well, thank you so much for the introduction, and thank you for the opportunity to be here. And I just wanted to say thank you to your company for investing in this cancer that so many of us, have really dedicated so much time to as well. This is really an underserved population in terms of access to trials and drugs, and it's just great that finally companies have really woken up to understand that there's a huge unmet need here. And so thank you for all the work that your company is doing. So next slide. These are my disclosures. And I guess this is a fairly high-level talk, just going over what is a clinical trial. So next slide. So, this may be a little bit basic for some people on this call, but when we think about drug development, traditionally, there are sort of three phases of drug development before a drug becomes widely available and approved for widespread use in a population. First, I should just say that before a drug ever goes into patients, it's studied usually in laboratories and in preclinical models, including mice and sometimes other animals, to understand how the drug works and to make sure that it has reasonable activity and safety. And then normally what happens is there's a first-in-man study where the drug is brought forward into patients for the first time, usually at a very low dose, so usually a fraction of the dose that caused side effects in animals, and then slowly that dose is ramped up. Traditionally, phase 1 trials actually happen in healthy volunteers, traditionally. And then after a safe dose is established in patients in a phase 1 trial, traditionally then there are phase 2 trials that for the first time try to get a sense of efficacy. Does the drug actually work to cure or treat disease? And then finally in phase 3, the drug is directly compared to whatever is the current standard of care for that disease or for that cancer type before it's FDA approved, to really show that it's better than what else is out there. Now, I have to say, this is a little bit overly simplistic and no longer the only way that drugs are developed. And I would say that in oncology in particular, where there's really a patient need, often multiple stages of drug development are combined to try to speed up drug development. And so we actually have, for the first time, drugs getting approved earlier, not necessarily after randomized phase 3 trials. Oftentimes, the first-in-person studies happen not to healthy volunteers, but to people with cancer. And so a lot has changed, but this is a rough framework of how this can work. So next slide. And so, I would say phase 1-2 studies are actually pretty much the norm in oncology now, where we try to get a sense not only of safety, but also of some efficacy in the initial studies in patients. So next slide. Next slide. So this is an overview of cholangiocarcinoma. I think my guess is that people on this call are fairly familiar with this, but I think it's helpful to just give a broad framework. So cholangiocarcinoma, sometimes also called biliary tract cancers, are cancers that start in the bile ducts. They either start in the liver, in the bile ducts within the liver, or the bile ducts outside of the liver. And oftentimes when we see patients with cholangiocarcinoma, we try to subtype them anatomically. So we call them intrahepatic cholangiocarcinoma, so tumors that start in the liver, or extrahepatic cholangiocarcinomas, which start outside of the liver. This is considered to be a relatively rare cancer. It's about 10% of all liver cancers are cholangiocarcinoma. These cancers are a little more common in other parts of the world, particularly Southeast Asia, where often patients who have cholangiocarcinoma have an antecedent history of liver fluke infection, and those flukes irritate the bile ducts and cause cholangiocarcinoma. That's much less common in the sort of patients that I see here in Baltimore, Maryland. So next slide. And I think the reason that I'm making a distinction between intrahepatic and extrahepatic cholangiocarcinoma is that these are, even though they're grouped together as one tumor type, there are some differences in intrahepatic versus extrahepatic cholangiocarcinoma. And in particular, the mutations in those cancers can be different. So there's some mutations like IDH1 mutations and FGFR2 fusions, which tend to be found almost exclusively in the cholangiocarcinomas that start within the liver. And conversely, for the cholangiocarcinomas that start outside of the liver, those cancers are more likely to have KRAS mutations or TP53 mutations. And so these cancers are not only anatomically different, but often have biological differences. So next slide. So where are we in terms of treating cholangiocarcinoma? This is standard of care in a nutshell. And so I would say for patients who have advanced stage cholangiocarcinoma, the standard of care today is to use gemcitabine cisplatin with usually a PD-1 or PD-L1 checkpoint inhibitor. So gemcitabine cisplatin durvalumab or gemcitabine cisplatin pembrolizumab. And this is actually a relatively recent advancement. So these triplet combinations are all new from the last 2 years. And then if patients either become intolerant or stop responding to the first line options, in the second line, for the majority of patients, we offer additional chemotherapies like FOLFOX or 5-FU liposomal irinotecan or FOLFIRI. Or for the subset of patients who have particular mutations that we have drugs for, those patients often get targeted therapy in the second line setting. So for example, for patients who have an IDH1 mutation, we might consider an IDH1 inhibitor in second line. For patients with an FGFR2 fusion, we might consider an FGFR2 inhibitor in second line. So that's really where the standard of care is today. Again, a lot of activity just in the last couple of years. A lot of our drugs are actually pretty new, which is very exciting. And we hope that, the next time that I come back for a webinar, we'll have a lot more to report. But this is where we are today. So next slide. So for those of you who know me, I spend most of my life actually doing cholangiocarcinoma research. And, one of the things that I am particularly interested in is the interaction between the genetics of cholangiocarcinoma and the phenotype of cholangiocarcinoma. How do the genetics of the disease impact, in particular, the immune system? And so this is data from hundreds of patients, over 400 patients with cholangiocarcinoma who underwent molecular testing with an assay called Tempus to figure out all the mutations in their tumor. And what you can see is that there really are distinct sort of subgroups of cholangiocarcinoma. So all the way on the right, the group that is green there, the mutational cluster 4, these are patients, as you can see, that predominantly have FGFR2 fusions or rearrangements. That's a very distinct group of patients. And then on the left here, you can see this is a group that has more TP53 and KRAS mutations, which is really a different phenotype of this cancer. So next slide. And in particular today, we're talking about this Genfit clinical trial. And so I think it's helpful to really understand a little bit about KRAS biology, because this in particular is the subgroup of patients who are hypothesized to potentially derive benefit from the drug that Genfit is developing. So KRAS is actually the most common mutation in cancer in general, not specific to cholangiocarcinoma, but any cancer. And so KRAS is a central driver of many different types of cancers, about 90% of pancreas cancers, and is actually very frequent alteration in cholangiocarcinoma. It's probably about 20% of intrahepatic cholangios and about 40% of extrahepatic cholangiocarcinoma. And KRAS is something that turns on cell growth. And so when patients have an alteration in KRAS, for example, a G12D, V, C, or D mutation, just for example, these mutations lead to KRAS being turned on that leads to activation of downstream molecules like MEK and ERK, which ultimately lead to this pathway of uncontrolled growth. And this is actually one of the first mutations that happens in cancer. So it's really a cancer driver. And in fact, cancers often can't live without KRAS. They're sort of addicted to this pathway. There are actually, for the first time, some KRAS inhibitors in development, which is an exciting area of development in oncology. But those inhibitors, even when they work, they don't tend to be curative. They just sort of control cancer for a period of time. And so I think we really need to, as researchers, think about how do we control KRAS-driven cancers, and are there specific sort of vulnerabilities that KRAS sets up for the cell? So next slide. And so again, this is the same pathway here. When RAS is turned on, it leads to activation of RAF, which leads to activation of MEK, which leads to activation of ERK. And a consequence of this dysregulated pathway, this RAS-RAF MEK-ERK signaling pathway, is that it turns on something called autophagy. Autophagy is basically a cell survival pathway. And so it's one of the reasons why cancer cells are particularly hard to kill, is that they turn on these pathways that prevent cell death. One of the things that I believe, and this is research from many groups, is that when autophagy is turned on, it actually leads to sort of a synthetic lethality or a particular vulnerability to treatment with an autophagy inhibitor. And so the overall hypothesis here that Genfit is testing with their clinical trial is for these tumors that have this activation of RAS, RAF, MEK, and ERK, can we block MEK, which leads to this compensatory upregulation of autophagy, but then can we come in with an autophagy inhibitor? And so can the combination of a MEK inhibitor and an autophagy inhibitor lead to arrest of cell growth and actually death of the cancer cell? And so that's really what we're trying to study. This has been reported in mice, and there's actually a case report of a patient with pancreas cancer who was treated with this combination, who had a phenomenal response. But this idea has not been tested before prospectively in a group of patients, and certainly not in a group of patients with cholangiocarcinoma. Next slide. So autophagy is something that many people have tried to target for a long time. But one of the challenges is that a lot of the autophagy inhibitors that have been brought into the clinic have had pretty limited activity. And there just have not been great autophagy inhibitors. GNS561 is a very, very potent inhibitor of autophagy. And it was actually studied already in a trial that included patients with liver cancer, where even as a single agent, it had some meaningful activity. And so this is already has some evidence of activity by itself. What has not been studied is when you give a MEK inhibitor and you drive up autophagy, does that lead to even greater benefit from GNS561? That has not been tested before. So next slide. So these are the results of the first in-person study of GNS561. And I just want to be clear, I had nothing to do with this trial. This trial was done by a number of other investigators. But what you can see is that in a group of patients who had pretty refractory cancers, so most of these patients had multiple treatments already for their cancers and were really out of standard options. And it was a mix of patients with pancreas, colorectal, and cholangiocarcinoma and HCC, there were patients who had disease stabilization, including a couple of patients who had some minor shrinkage of their tumors, including 2 with intrahepatic cholangiocarcinoma. So, when I look at this data, I don't think that this was a home run by any means, but it does suggest that there's something to this autophagy inhibitor, that it has some activity. And so really the question is, can we make this work better, understanding the biology of autophagy? So next slide. So the idea is, can we block MEK, turn on autophagy, and then come in with this autophagy inhibitor? So how do we block MEK? The good news is that there are already MEK inhibitors in the clinic. There are a couple of them, cobimetinib, trametinib, among others. And these are very specific drugs that block MEK. They're approved for multiple cancers. And I want to mention that MEK inhibitors have actually also been studied for cholangiocarcinoma in the past. I led a study of cobimetinib plus a checkpoint inhibitor in cholangiocarcinoma, and there is some activity to MEK inhibitors already in cholangiocarcinoma, although again, not a home run as a single agent in cholangiocarcinoma, but there's some activity. So next slide. So this is the design of the phase 1/2 study, and I think I've already hinted at exactly what the design is, but we have an autophagy inhibitor. We have trametinib, which is a MEK-1/2 inhibitor that is already used for many cancers. And so the question that this clinical trial is asking is, if you give both an autophagy inhibitor and a MEK inhibitor, is there real synergy? And does the combination lead to inhibition of tumor growth? So next slide. So this is an active study. It's listed on clinicaltrials.gov. The sponsor is the company that is putting together this webinar. Carol is here on behalf of the company. I'm really here as a researcher with interest in this pathway, and I had some involvement in how this trial was put together. This trial is only for patients with KRAS-mutated cholangiocarcinoma. So I just want to make that clear to a lot of the patients on the webinar who might be excited about this. Right now, based on how we think this is going to work, the trial is only open to that 20 to 40% of patients who have a known KRAS mutation. So patients who don't have KRAS mutations, this study is not an option at this point. It's for patients who have really run out of standard first-line options, so patients can get on in later line setting. And the 2 drugs are both oral pills, and they're given together. And patients can stay on this as long as the pills are working and as long as patients are having some success without unacceptable toxicities. So next slide. So this is really the study design. And again, as I mentioned earlier, in oncology, we often, given the patient need, try to get drugs through development as quickly as possible, which means combining phase 1 trials with phase 2 trials. And so that's really how this was set up. There's first a dose finding aspect of the study. Fortunately, we have a safe dose for trametinib already in patients, and we have what we think is a good dose of the GNS561, but because this is the first time that these 2 agents are being combined, we want to make sure that there's not unexpected toxicity combining these 2 drugs. So there's first a sort of dose optimization portion of the study. And then after that, there's an expansion where really the goal is to understand at that optimal dose, is there some evidence of real meaningful clinical activity here? And so the major goals are, how safe and tolerable is this combination? And then is there evidence that this actually is working the way that we hope it works in terms of controlling cancer? So next slide. So where is this open? So you can see here, we're really excited that the study is now open at a couple of different sites with multiple other centers opening around many parts of the United States. Now, I have to say, it always makes me sad looking at these maps of the United States that there's nothing in the sort of middle of the country where at least one person here, I know Melinda hails from the great state of Montana, is that right? So, I think this is a real challenge when you're dealing with a rare cancer, and then it's hard to find studies, and I hope that we can do better. But at least multiple sites and hopefully an opportunity for a lot of patients to have access to this trial. Next slide. And so these are the sites that are open or close to opening. I'm here at Hopkins, where we're really hoping to join the study in the phase 2 portion. So next slide. And I guess that actually brings us to the end and time for questions. I hope I didn't go over my portion.
No, not at all. So I invite the audience to go ahead and throw questions in the Q&A or in the chat box that you have for Dr. Yorshuan or for Dr. Addy, and we will get to those. I know I had a few, so I'll start, and I think you might have answered it along the way, but why has it taken so long for researchers to be able to target KRAS, and why is it so hard to be able to do that?
Yeah, such a good question. There's a couple of different answers. Every single grant that I ever wrote that had anything to do with KRAS, the first sentence was always, "This is a completely undruggable driver gene." And so, I think it's a really good question. We have IDH1 mutations, we have an IDH1 inhibitor. We have FGFR2 fusions, we have an FGFR2 inhibitor. We have PIK3CA mutations, and at least in other cancers, we have PIK3CA inhibitors. Why do we not have a standard KRAS inhibitor? So they're actually, the biology of KRAS is really challenging, and it's just been very, very hard to develop a true inhibitor, but there actually have been breakthroughs. So just very recently, there was a KRAS G12C drug that was approved predominantly for patients with lung cancer. And there are other KRAS drugs coming that directly target KRAS. But I don't think our work is done at all when we look at these trials, even though they're very exciting, the KRAS inhibitor trials. And by the way, I'm totally enthusiastic about KRAS inhibitors. I think this is a breakthrough. But I think the challenge is that we do find that resistance develops. And so patients who go on KRAS inhibitors, normally, even if they work in a matter of months, often the cancer finds ways to kind of escape blocking KRAS directly. And so I think even if we have KRAS inhibitors for cholangiocarcinoma, and I hope one day we will, I think there's still going to be a need for other ways to target KRAS-activated cancers, including pathways downstream of KRAS that should still be activated even if the cancer finds a way around a KRAS inhibitor. So, I would just say, we do have a lot of progress drugging KRAS, but a lot more work to do.
Thank you, and good. Sounds very hopeful. So the first question is, what is the timeline for the second portion of the study and the upcoming locations? Will they be the same?
I will turn those questions over to Carol, who has the best sense of, so she's with the sponsor about where the timeline is.
Thank you, Melinda. So where we are currently, we enrolled the first patient relatively recently. So we're slowly progressing through phase 1B of the trial. And as you pointed out, Dr. Yorshuan, this part of the trial, because this is the first time that we are investigating this combination, we're taking a very slow and cautious approach. And what that will mean is doing very slow and careful analysis of all of the data as we accrue patients in that portion of the trial. So right now we're anticipating approximately 20 patients in phase 1B, after which we will be looking at all of the available data so that we may inform the doses that will then be carried forward to phase 2A. So our hope is that we will be getting there probably 2025, but again, it will be really dictated by our experience as we move forward with phase 1B and we slowly and gradually escalate the dose of GNS561, and this will all really be driven, as you pointed out, Dr. Yarshowan, by the safety and tolerability that we're seeing, how patients are doing with this combination.
Okay, and then maybe another question for you, Dr. Addy, is do you expect any studies to start in Europe or sites to be opened in Europe?
Currently, as we outlined on the slide, all of our sites are anticipated to be in the United States. We are, however, exploring other geographic areas. So the best way to stay up to date is to just follow on clinicaltrials.gov, because as soon as we open new sites, new countries, we will be posting those on the website.
Okay, perfect. And is this study for all KRAS variants? So Dr. Yarshowan, you mentioned G12C and D, so would it encompass all of those?
Indeed.
Yeah, any KRAS activating mutation. So the most, for whatever reason, that position 12 in KRAS is the most common. So KRAS G12D, G12V, G12C, G12R, G12A, but actually any KRAS activating mutation, G13 happens sometimes. Any of them are included.
Perfect. I love to hear that. If you've already tried a KRAS inhibitor previously, does this disqualify a patient from the Genfit trial?
I think we have a specific exclusion for previously having had MEK-inhibitor. But I think at the point in time, Dr. Yarshowan, as we work together with the concept of this trial, I don't think yet that the KRAS inhibitors had hit the radar screen.
I think what patients would be worried about is maybe already participating in a previous clinical trial with a KRAS inhibitor, if they would be excluded from doing this trial as well. Nope.
Not to my knowledge.
So just the MEK inhibitor.
Yes.
Okay, perfect. Thank you. You answered the KRAS G12R mutation is also included in the trial. So as you mentioned, this is targeted towards patients with failure of first-line therapy. Is the trial only for patients looking at second-line therapy or at any point in treatment after first line?
This would be, Melinda, for any patient who has previously failed first line, so anything thereafter. So it could be second line or a patient who maybe had already failed second line. Those patients would also be eligible.
Yeah, second line and beyond.
Yes.
There are some, patients do need to have sort of adequate organ function. So platelet count above, we said 100,000, hemoglobin above 9, reasonable liver enzymes. So there are some other important criteria, but the number of lines of therapy is not capped.
Okay. Okay, could you speak on the difference between a phase 2A state versus 1 and 2? So I think what they're talking about is the phase 1, phase 2 trial. How does a 2A fit in there?
I think how we call a study is increasingly not important, I think. And I'll just give you an example directly from the cholangiocarcinoma world, which is when patients progress on first-line gemcitabine cisplatin-based therapy, we basically have two standard of care options for second line for patients who don't have an actionable driver. We have FOLFIRI and we have FOLFOX. FOLFOX was a phase 3 trial. FOLFIRI was a phase 2 trial. But if you look at the number of patients, it's basically the same. So I think we can call these studies whatever we want. And the names of what we call them is not always telling you much about what the study really is. And so I actually want us as a community to sort of move away from the emphasis on what we call the phase and really focus on what was the goal of the study and how many patients were on it. And in this study, I think, I think we could call it a phase 1, 2A, but I think it's basically Is it safe? And is there some hint of efficacy that makes us want to continue this? And so that's really the goal here.
I like your way of thinking.
I agree. I agree with that. And, you know, I also agree that what we call trials is in many ways artificial. And I think when we think about the trial that we are conducting, certainly we don't debate that there are two parts, but those two parts have very distinct goals, right? So the first part is let's evaluate the safety of the administration of these two investigational drugs so we can pick the optimal dose that we carry forward into the second part. And that is the part where we will really truly begin to evaluate how it's working.
Yes. Well, I like your way of thinking. I always tell patients to not hesitate over the phase. And I think they're all potentially beneficial, and they're all beneficial for the science to move forward. Okay. Can you say anything about adverse events that have been seen with the GNS 561? Or what would be the anticipated side effects that you're thinking could happen?
So I can speak to this for a sec. There was a initial first in human study that was done by my friend and colleague James Harding at MSK. Looking at the data, so I did not participate in this study, but I think the drug was actually pretty well tolerated. The most common adverse events were sort of gastrointestinal adverse events, so some nausea, vomiting. In a subset of patients, about half of patients had some kind of GI upset. There was some diarrhea. And then some patients had some liver function abnormalities and some fatigue. It's always hard to know. These are patients with cancer. Are we seeing side effects of the cancer rather than the drug? But overall, it was a relatively favorable safety profile. The trametinib, I think also is worth just talking about. So trametinib is approved. There's an FDA label that any patient could pull up to read what the side effects are, but it does have some abnormalities in certain muscle enzymes. It can cause some fatigue. It can cause actually some fevers, which is a real problem for some of our patients. But overall, also not I think reasonably well tolerated in my experience. I put a lot of patients on these MEK inhibitors.
Okay, good to hear. So what do you think about the possible use of a RAS inhibitor for KRAS mutated patients? Does that make sense?
So I may be misunderstanding the question. So for a patient who has a KRAS mutation, there are direct inhibitors of KRAS signaling, so KRAS inhibitors. There are also kind of pan-RAS, RAF inhibitors that were developed in the past that didn't really show activity. I do think that the new KRAS inhibitors from companies like Amgen and Revolution Medicines and recently now BMS, who bought Mirati, these are showing some meaningful activity, and I think it's very, very exciting. So more to come there.
Okay. And Dr. Heidi, I don't know if you can share the slide with the sites on it. Someone's asking, will Memorial Sloan Kettering be involved with clinical trial testing?
We have not yet had connection with Memorial Sloan Kettering, but as you see, we do have a handful of sites that are in the Northeast, so that would be, possible. We do have a patient concierge service with whom we are working to enable patients who have to travel some distance in order to get to clinical trial sites. So I think that's important to understand. So travel accommodation support is something that we look to support patients who are interested in participating.
Love to hear that. Thank you for doing that. We appreciate that. Are peritoneal mets an exclusion?
Not to my knowledge. Carol, can you back me up?
Not to my knowledge as well. We would have to confirm that, but not to my knowledge as well.
Okay, some off topic. So this is what the study is showing. The question is it anticipated that some patients will have their cancer shrink to allow resection? But that is part of the, I guess, the 2A, the efficacy, to see what kind of efficacy we would come from this, right?
You know, I do my job and I love my job because I have optimism that we really are starting to have really effective therapies. And I certainly hope that this drug combination will shrink cancer. And for patients who have unresectable disease, there's nothing more satisfying as a doctor to get patients to potentially curative surgery. But you know, that said, I also want to be realistic that we really don't know if this is going to work. And you know, that's why we do trials. The one thing we always promise patients when they do trials is that they're helping other patients, because you know, the only way that we advance the field is by trying new things.
Absolutely. The other thing I tell patients is that they're so closely monitored, and you know, it's definitely worth considering clinical trials as a treatment option. Okay. Can you explain any major difference between durvalumab and pembrolizumab and their efficacy that you touched on the standard of care treatments?
Absolutely. So I would just say from a clinical perspective, the data looks very similar between the two. And so, you know, I've had patients ask me which one is better, and the truth is, I think they're probably very similar clinically. They are a little bit different in terms of, you know, how they work. So durvalumab is a PD-L1 inhibitor. Pembrolizumab is a PD-1 inhibitor. So PD-L1 binds to PD-1. So these antibodies block the opposite ends of the equation, basically. But they have the same net result of blocking the interaction between PD-L1 and PD-1. I think as an immunologist, I often was under the perception that blocking PD-1 might be better, because PD-1 also binds to PD-L2, which is another interaction. So if you block PD-1, you block PD-L1, PD-1, and PD-L2, PD-1. Whereas if you only block PD-L1, you don't block that second co-inhibitory interaction. But that really hasn't borne out with the clinical data. I think the clinical data is very similar or equivalent. So I think both are perfectly reasonable options. And right now, unfortunately, it's kind of a coin flip about which one we use.
Thank you. So if someone were to join the trial and the treatment was working, if the trial ended, can the treatment be continued?
Yes, this would be something that we would make available. We could make the GNS 561 available, as this is in-licensed to Genfit. Trametinib, of course, is approved for other therapies or for other indications, and would not be provided by Genfit.
Okay.
But could be used off-label, of
Off-label.
course.
Okay. Can you please show the site names again? I think you had one slide with all the there. Yeah, that one. Perfect. And we talked about how many numbers you're looking to accrue 20 across all those sites.
Approximately 20 for the first part of the study. We won't call it phase 1B, the first part. And again, the total number will be dictated by how tolerable the combination is before we move into the second part of the study once we've identified the optimal doses.
Okay, thank you. What would be the best way if a patient is interested in participating in the trial to reach out?
There is a contact information in the clinicaltrials.gov site. So that's what I would encourage, is to reach out through the clinicaltrials.gov website. We will also have members of the Genfit team at the Cholangiocarcinoma Foundation Annual Conference next week. So if anyone will be attending in person, in fact, we will have a poster presentation that provides a little more detail about this trial. And so we would welcome you stopping by to pay a visit to learn more.
Well, perfect. That is a wonderful note to end on. Thank you for pitching the conference, because I invite everyone to attend next week as well, in person in Salt Lake City, the 17th through the 19th, or please join virtually. If you can't come in person, you can register to watch all the presentations virtually. And thank you guys so much for the webinar today and for all the information. This will be recorded and hosted on our YouTube channel. If you want to go back to watch or share it with anyone in particular, feel free to do so. And if you have any other questions, reach out to us and I will try to pass them on. Yes, I am confirming that there would be 20 patients in the initial portion of the trial. That was the last question. Any other questions, pass them on to advocacy@curecca.org and we'll get them passed on to Dr. Addy or Dr. Yorshuan. Thank you guys so much. Have a great day.
Thanks for having me.
Melinda, thank you. Thank you so much. Thank you, Dr. Yorshuan.